Hi everyone. I hope you all have been doing well in the biggest health emergency of the world, which is this pandemic COVID-19. I wish you all to be safe and to be at home and follow and obey all the guidelines from the authorities time to time. Uh, as there is a saying, the show must go on, and so our studies must go on. I am here with my lecture on the topic of irreversible cholinergic inhibitors. As you all must be remembering that we have finished or we have been through the classification of cholinergic drugs, that is also known as cholinomimetics, which was basically divided into two parts: direct acting agonist and indirect acting agonist. Amongst the direct acting agonist was, what were the things? Let me remind you. Those were acetic acid cholinesterase, carbamic acid cholinesterase, quinacridine derivatives of acetylcholine, and plant alkaloids. Amongst indirect acting agonists, it was divided basically into two parts. One was reversible, which consists of isostigmine, neostigmine, etc. And the other part is irreversible indirect acting cholinomimetics. Our focus in this lecture is on irreversible inhibitors. Irreversible inhibitors basically belong to a class which is organophosphates. As the name itself suggests, these are the organic compounds having phosphate group. We will see some examples of those. So organophosphate agents are typically used as insecticides, as well as for they were used in many wars. They were used as warfare nerve agents. They were used to paralyze or kill the soldiers of the enemy country. These compounds basically act as irreversible acetylcholinesterase inhibitor. If you remember, during the neurotransmitter cascade, during the neurotransmitter phases, what I told you, there is a very important neurotransmitter called acetylcholine. Acetylcholine comes to the receptors and it induces that receptor for further contraction of the muscles and various roles, as you are aware. But these organophosphates and those neurotransmitters later on are metabolized with the help of acetylcholinesterase. But here, when acetylcholinesterase is inhibited, and that also irreversibly, that means for longer period of time, maybe for hours or maybe for days, in that particular moment, there is a lot of acetylcholine concentration at the receptor, and it gives a lot of neuronal firing that can give seizures, respiratory arrest, and death. So this is sort of poisoning. Organophosphates are sort of poisons, you can say. These compounds act as irreversible acetylcholinesterase inhibitors and are effective in very low concentrations and can cause death within minutes to exposure. Acetylcholinesterase break down the neurotransmitter, as what I just explained you. They break down the neurotransmitter acetylcholine at neuronal synapses and neuromuscular junctions. And this irreversible inhibition can cause the neurotransmitter to accumulate in the synaptic cleft, leading to overstimulation of cholinergic receptors, which can cause seizures, respiratory arrest, and death. A general formula for such compounds as shown here, phosphate compound P is there. It can attach to X. X is a very good leaving group. It can be fluoro group, cyanide group, para-nitrophenoxy, or thiomalate group. A is either oxygen, sulfur, or selenium. As I told you, they are biosimilar groups. It can have those biosimilar groups, maybe oxygen, sulfur, or selenium. R1 and R2 can be alkoxyl group. R2 can be alkyl or tertiary amine as well. So why they are so strong binders that they are reversible? There is one reason listed here. When A other than oxygen, for example, phosphorus to sulfur, if there is A other than oxygen, that means there can be sulfur or then there can be selenium. So there is a coordination bond between phosphorus and this compound. Acetylcholinesterase inhibitors need to be bioactivated by cytochrome P450 enzymes to the corresponding phosphoester group. Why this is needed? That is, the PS is not as electrophilic as PO, that to allow nucleophilic attack by serine. We can understand it from here. This is organophosphate, but this is not very active. This remains inactive. It needs to be converted into this one. This happens with the help of oxidation inside the body, which is carried out by this P450 enzyme to convert PS into PO to allow nucleophilic attack by the serine. As you know, serine OH, if you remember, this is present inside the active pocket. As you remember, if you remember the mechanism of acetylcholine, this serine OH is present inside that active pocket of the receptor. Since insects appear to have more reactive oxidative enzymes, some selectivity is observed for these agents. Here you can see this is inactive, oxidation is made. So this by the replacement or intermediate changes, it comes S and O. S is then leaving and it forms active organophosphate group. The acetylcholinesterase inhibitory activity of PS, yeah, that means the organophosphate which contains S or selenium is 10 to the power 5-fold weaker than PO group. That is why inside the body they try to convert as PO to make it more powerful electrophilic agent. Now let's see how many types of organophosphates are there. The type has been classified based on the groups around phosphorus. Like number 1 is phosphate. Phosphate, you know, PO4. Around phosphorus there should be PO4. 1, 2, 3, 4. 4 oxygens are there. This is phosphate group. One example is para-oxon. You will see in the later slides. Another example is phosphothioate. Like here, what happens, as the name suggests, thioate, one oxygen is replaced by sulfur. In place of oxygen, there is sulfur. Another example. Third example is phosphorodithioate. Yeah, that means there should be 2 sulfur in place of 2 oxygens, as you can see here. And this class has a very well-known insecticide, malathion. Malathion and parathion are very well-known insecticides. We will see in further slides also why they are important. Fourth type of organophosphate is phosphonate. Here, one oxygen is replaced with carbon. Carbon is the part, and they can further be related to the different alkoxyl groups. The last one is phosphoramide. Phosphoramide, as you know, CONH2 is amide. So phosphoramide is PON and 2 substituents of nitrogen. Now let's see some examples of organophosphates. As I told you about nerve gases. Nerve gases have been used by the countries for warfare. Even in 2001 in US, some terrorist groups have used these nerve gases. Serine is one of the diflos and serine. They are notorious, infamous nerve gases. The structure, you can see the structures. This one is fluorodiflos, diisopropyl fluorophosphonate, and this is serine. These agents were developed in World War II. They irreversibly inhibit acetylcholinesterase. They make permanent activation of cholinergic receptors by acetylcholine. Permanent activation, that means it can create excessive neuronal firing, which can give convulsions, which can give cardiac arrest, which can give death as well. Now, let's see the mechanism of action, how organophosphates work. As in the previous slides, I told you there is serine OH in the active pocket. We will start from there. When organophosphate reach over there, the lone pair electron of phosphorus transfers on fluorine, and there is plus charge. This bond, fluorine goes out. Fluorine takes that bond and it goes out. So phosphorus develops one positive charge. One positive charge because it's donating its lone pair of electron. So there is lone pair of electron with the serine OH. This lone pair of electron will be transferred to this, and this bonding between O and P is very strong. That is why this type of organophosphate bonding and serine, they are irreversible. They do not replace so easily. They have very long T-half. It takes time. That is why they are very strong poisons and they are stable. So that's what I just said, irreversible phosphorylation. TO bond is very stable. There is another example of DFP, difluorophosphate with acetylcholinesterase enzyme. You can see just here, they have made an intermediate, and for very long time, it remains as inactive acetylcholinesterase while it do not converts, it do not leave it, and it let not native acetylcholinesterase remain in active form. There are certain, until now, I have been just talking that organophosphates are insecticidal, are poisonous, but as these are acetylcholinesterase inhibitors, so they have some medicinal value as well. So this class is medicinal organophosphate, and the example is ecothiopate. Ecothiopate has this structure. It contains, as you can see, it contains quaternary nitrogen group, which improves the binding interactions, and moreover, it results in better selectivity in lower and safer doses. This is used to treat glaucoma, and it is applied as topical application. This is not given into the systemic application. This is applied just for topical application. Next is Let's see organophosphates as insecticides. How do they act in insects? As the two major examples are parathion and malathion. The structures you can see, these are relative. There can be a question. There should be a question coming to the mind. These are insecticides, and these are acetylcholinesterase inhibitors, irreversible inhibitors. So they can enter into our body, and it can harm us also. It can be the reason for the death, or for seizures, or for cardiac arrest. But let me tell you, and we will see more mechanisms. Parathion and malathion are more susceptible into insects. They have very rare susceptibility for humans, until and unless in high doses. So they are relatively harmless to mammals until and unless they come into some considerable concentration. It does not harm. So they are relatively harmless to mammals. Agents act as prodrugs in insects, metabolized by insect to produce a toxic metabolite. Let's see how. Parathion in mammals and in insects. In insects, that parathion is an active drug, but this is an inactive prodrug. That same parathion is inactive prodrug in the humans if ingested. In insects, this phosphorylase enzyme, acetylcholinesterase enzyme, phosphorylates means it creates the bonding, PO bond, and inhibit acetylcholinesterase enzyme and create death. But in mammals, in mammal metabolism, it is inactive and excreted. It does not show effect into the human directly. Malathion is the least toxic organophosphate known till date due to the presence of ester functions in its molecule. It is generally known that insects have much lower esterase activity than the mammals. What does it mean? It means that this drug malathion has ester group inside, and inside human plasma there is esterase which breaks this one and creates non-toxic metabolite. This you can see, the metabolite is non-toxic. But in human, the esterases is not very high but in insects the esterases is not very high. So in insects this is toxic. It is not metabolized like this, so it is toxic. It works as insecticide, but in human this is safe. So this was about malathion and parathion. They are very important insecticides. Sorry. Phosphoramide. And there was one class known as phosphoramide. Sorry. Phosphoramide, as you can see, is a type of organophosphate, but this is inactive due to the absence of a good leaving group within the molecule. Their selective toxicity is realized due to the differences in the metabolic biotransformations between insect and mammals. In human, this is converted into an inactive metabolite, but the same compound inside the insect is converted to an active metabolite. By this way, they are successful insecticides. Now we have been talking about the thing that it is like a poison, reversible. Anyhow, if there is organophosphate poisoning in humans. So what are the ways to go out? There is a thing called reactivation of acetylcholineesterases. The acetylcholineesterase, which has been um, poisoned, we need to reactivate it. For that, there is a strategy. There should be a strong nucleophile required to cleave that PO bond and find suitable nucleophile capable of cleaving that esteric bond. Water is too weak for that. Hydroxylamine could be a stronger nucleophile. So scientists thought in that way. Hydroxylamine, this could bind with that, and that can leave the serine part of the receptor. So it can be a drug, but hydroxylamine was too toxic for this use. It increased selectivity by increasing binding interactions with active site, but it was toxic. So for that reason, scientists developed pralidoxime chloride, better known as PAM. PAM has quaternary nitrogen, which helps to bind the anionic region. Side chain is designed in a way to place the hydroxyl amine moiety in the correct position relative to phosphorylated serine. It cannot cross blood-brain barrier due to charge, so it cannot act on CNS. It is used as antidote for poisoning by parathion and related pesticides. It is also effective antagonist against neostigmine. This is the mechanism how pralidoxime works. As you can see here, in the active blocked site, there is organophosphate. So what happens with the pralidoxime? The lone pair of electron, they attack this phosphorus, and this bond is broken down, and they can replace this hydrogen over here. This serine is now free. Active site is free. So now when active site is free, this can break down the acetylcholines. Excessive of neuronal firings can be stopped, and acetylcholineesterases is reactivated. So this has worked as antidote. The same way, there is another drug called propam. If you remember, PAM was not active in CNS. Acetylcholineesterases activity is also there in CNS. Because of that, there is a convulsion. So propam, another reactivator, this passes blood-brain barrier. So it can have effects in CNS. Propam has benefit over PAM. At last, the summary. Organophosphates are irreversible indirect acting cholinergic agents. They cause overstimulation of cholinergic receptors, which creates seizures, respiratory arrest, and death. It is used as insecticide and warfare agents, clinically used to treat glaucoma. The antidotes are PAM and propam. Thank you very much. For the questions, you can reach me through the emails. Thank you so much. Thank you.